Then, of course, to verify whether we have the correct genes, we need to make knockouts in the penicillium strain, and we managed to construct an important mutant which we have to use for constructing the knockouts, because we have to prevent them from reverting, and because there's this non-homologous end joining, and we have to knock out the gene which is helping to do this recombination. And this was done, so now we can start to create the knockouts. And so we can now see what the individual genes are really doing, whether they are really involved in the biosynthesis of cyclochlorotyne. Now, for the biosynthesis of the pyrrole moiety, which is necessary for halogenation, we need a pyrrole dehydrogenase gene. And in peptide biosynthesis, you need a peptidyl carrier protein, which you load, and you load the amino acid onto it, in this case proline. Then you have to oxidize proline to the carboxylic acid moiety and then do the halogenation reaction. So it's a multi-step reaction, and of course you need all these genes. And one of these genes is this dehydrogenase. And here you have an example of such a dehydrogenase. It's a flavin-containing enzyme, so it's yellow. This is a thermophilic one, which was used just to establish the procedure. And here we have one from the cyclochlorotyne producer. The enzyme, it's one of 3 putative enzymes, and one was purified and it's active. So now we can use these enzymes to work on. And coming to the second step here, the halogenation, after the reduction of the proline, of the oxidation of the proline, we need a halogenase. Now with these halogenases, we have the situation that there are a number of these flavin-dependent halogenases are known, and they use different substrates. Now to detect the correct gene, we need primers which are specific for these pyrrole halogenases. So we cannot use the tryptophan halogenases as primers and probes, and what we could do is construct specific primers and probes so we could distinguish a halogenase, and we also found it in the genome. We could purify the enzyme, but the problem with showing activity is that we need, of course, now the peptidyl carrier protein bound substrate. And since we could not show activity using a chemical synthesized mimic, we decided to do a complementation experiment, because we have a mutant in our hands which has the halogenase deleted and which also works with the same system. And we made the constructs, we made the recombination, and so far we have not analyzed the products which came off because somebody got ill, and so we still have to wait whether the complementation was successful in this case, whether we really have the active enzyme. Now the question is, where are these astins in the plant? There are plants which produce astins, and there are plants which obviously do not produce astins. We've got an astin from Austria, which produces, and we got an aster from Russia, which is grown in the botanical garden in Dresden, which does not produce the compounds. And you can see, you can find them everywhere in the plant. They mount stiffer, but they are there in the plant, and the question is now, where do they really come from? Because of the similarity with the penicillium strain and the astins, the question is, are astins just derivatives of cyclochlorotyne produced by the penicillium strain, or are they actually produced by the plant? And we analyzed a number of penicillium strains, and they all cluster here, and they all produce cyclochlorothine, none of them is producing astins. So clearly it's not the penicillium that is producing the astins. Then we thought, could there be an endophyte present in the roots of the asters producing astins? So we analyzed plants which were surface sterilized, then we looked for fungal endophytes, isolated them, then also characterized them and extracted compounds from them, but none of these endophytes produced any astins. So our assumption that astins are produced by the plant itself seemed to be correct. Since we wanted to express the astin genes, the biosynthetic genes, also in plants, hairy roots, we needed sterile plants. So plants were sterilized and grown, but in the end, these plants turned out not to be really sterile. Because when they were grown, and here you see, it took 2 months to really see what happened, then suddenly a pink fungus popped up. This fungus was isolated, and it was a novel fungus, not known before. And when we analyzed the biosynthetic properties of this fungus, we could establish that it was actually this fungus that produces the astins. So the astins are not produced by the penicillium strain, they are not produced by the plant, but they are produced by a yet unknown fungus. But not all the astins which are known could so far be isolated from the fungus. So there's of course the question, is the plant also doing something to make derivatives of the astins? Question mark. Now just something for the relationship between the new endophyte and the penicillium strain. They're not really related. They fit on the same screen, but they're not close together. Also, it's quite interesting that the compounds are rather similar. So this is evolution. Of course, we now sequence the new fungus, and I just show you one slide here comparing the astin-producing endophyte with the penicillium strain. The genome is smaller than in the penicillium, GC content is higher, and also you see the penicillium strain is producing more secondary metabolites, or has more genes for secondary metabolites than our new astin producer. And of course, the question now is, what is this fungus doing? Is it just producing the astins, or is it also producing other compounds which are used in the root extracts of the plant? Of course, I cannot show you details of the sequencing data from the new fungus, but as a last slide, one slide before the last one, I can show you comparison. And since we have the 2 candidate gene clusters from penicillium strain, and we have 1 candidate gene cluster in the astin-producing fungus, there are close similarities with the genes we need for the biosynthesis themselves, genes we need for precursor production. And with the last slide, I would like to show you a comparison, showing what we wanted to do. Then the slight detour using the cyclochlorothine producer, meaning we wanted to isolate the genes, put them together, do heterologous expression. For this, we decided to use the penicillium strain to get these genes, but in the end, it was much easier because all we needed was the new fungus. There's everything in there. So we have an expression, we have the compound, and now it's as easy as that, just ferment the strain, produce the compound, and now we can do the analysis of the compounds, looking for novel activities, and now we need a few liters to get the same amount as you can get using kilograms of astin roots. And with this, I'd like to thank you for your attention.
Great, thank you. Any questions, please?
Did you check any other ester species for the presence of this fungus, or is there any idea where the fungus might live elsewhere?
So far, we have not checked them for the fungus. We plan to do this because now we have the genome, so we have probes and can really look in the plant themselves, but we have to make constructs to really see where the fungus is sitting. This should be possible using fluorescence. And so far, we've not seen this fungus anywhere else. It did not pop up in the roots. So it seems that it's present somewhere inside the plant, and because the compound is extracted from the roots, seems to be due to diffusion of the compound throughout the plant.
And are there any approaches to take the genes that you need for the ester production out and put it into some other, whatever, Saccharomyces or?
This is something we were thinking about, because now we have a new system, a known system, but not well investigated. You see the wording of the ERA-IB-5 call being repeated here. And of course, the question is, can we now use this strain as it is for a high producer? Or will we get better results by taking out the genes, putting them into another strain? E. coli, definitely not the choice, because this would certainly not work for such a system. It could be plants, but then we're back into plants. It could be yeast. So the question is open what we can do, but one important thing is, of course, to improve the production using this fungus.
Question at the back.
What is molecular target of astin?
In literature, you can find that astin is an activator of caspase. So you have a more general apoptosis induction.
Thank you.
Any more questions? No, let's thank the speaker. Thank you. Okay, so again, we're good on time. We've got, I think lunch we've managed to push forward to 1. So we have a question. I have a question for you before we decide what to do next. How many of you in the room here might be looking for partners for the next call or the current call? Any hands up? 2, 3, 4, 5. Here's an alternative. I'll look over at Karen and see what she thinks. Would those people in the room who are looking for partners like to have a minute or two to say what who they're looking for, or the skills or techniques or capabilities they might offer? Should we do that? You don't want to do that? No? How about anyone else? Would anyone else like to sort of do a 2 or 3 minute pitch about where they work and what they do and what they might be able to offer to other partners? Any more than one person? Anyone else? 2 maybe? Any more than 2? So if it's only 2, we can do another talk. What do you think? What do you want to do, Karen? Okay, we can be the 4. Yeah, we could do it that way if you like. Well, let's start with the 2 at the back who are interested, and then we'll see how if we can build a bit of momentum. So, Dilek, if you let these 2 gentlemen at the back just explain that what they do and what they might be looking for, and that should hopefully that will take us through to lunch. And then we'll have 3 presentations for after lunch.
Okay. Well, it's a favorite position here, right in the back. I should say come in front of you. It's better to listen, see. So what we can offer is reaction engineering. Currently, it's done by catalysis. is you have an enzyme probably does not work, and strategy which is mostly pursued is mutagenesis, enzyme engineering, and so on. But there is only a meager understanding of the processes inside the reactor, and the reaction engineering can indeed help to promote a process, but you have to do this work by the knowledge of a chemist, like I myself am, and we can also offer the opportunity to do some large-scale experiments up to 20 liters, so we can produce on the kilogram scale. We have a small mini plant, and if there is a project that could profit from having a chemist aboard with has got some knowledge in reaction engineering, can do some large-scale experiments, I will be happy to join you. Thank you.
Okay, so my name is Nicholas. I'm Nicholas Sita from University College London, Biochemical Engineering. I am here because we were funded, I think it was the 4th call, for a project where we're looking at microfluidic tools to do process integration, in particular the downstream processing side. And so I think that in a few years we will have to report on our achievements, so I thought it might be good to see how this is done. This is the main reason almost that I'm here, so I'm not really prepared to give a pitch on any projects that we're particularly looking for, but I'm very happy to sort of briefly say what we could offer in a project. From my group, looking at the microreactor and microfluidic side, I'm very interested to sort of expand that technology. We have built reactors for enzymatic conversions and looked into reactions on the flow in microfluidic systems. And now, as I said, sort of integrating these with downstream processing, unit operation steps such as, say, liquid-liquid extraction, where you get a real nice benefit from the miniaturization. As a department of biochemical engineering, and this is now a little bit similar to what my previous predecessor said, we're looking at a number of issues in getting the processes up to, translating discoveries to processes. So we can also offer facilities in upstream and downstream processing towards pilot scale. Currently our largest reactor is 400 liter, but we're probably decommissioning it for a smaller one because it's a bit expensive to keep these running, as I'm sure you know. So if there are projects in the area of process engineering, I'm sure there's a lot of people in my department that would be interested. And me personally is more on the micro side. One of the speakers mentioned synthetic biology, and I think this is also a very exciting field where a number of my colleagues now want to sort of get into. So I hope this has given you a bit of a roundup, and that's all. Thank you.